everyone for making a huge effort to be here today with us. We haven't really met together for so long for one reason or another, and it's wonderful that we can at least see each other on the laptop, on the computer, and hear about what we're doing and what's going on. I think it's been long overdue, and I'm hoping this is one of the last virtual meetings we have, and that the next meeting we all get together somewhere in the world and really share what we're doing, because I think it's a really important area of development. So as you'll see, we have an agenda, and we're going to have very short talks from everyone, which I think is just to really bring everyone back up to speed of where we are, what we're doing in our different biofoundries, and then we'll have a break in an hour's time, and then I think we continue that, and then we have some discussion at the end of the meeting about the the GBA, where we're going, what we're doing, and the future of what we're doing, and whether there's opportunities to work more closely together and collaborate. So that's a very open discussion for everyone to contribute. So really a really great welcome to everyone. I'm so happy that people made the effort to join us, and I'm really super looking forward to hearing what's going on around the world, in our different biofoundries. So I think it's a Zoom call, and I think everyone can share their screen, and I think I'm really happy to get going if that's okay. If anyone else wants to say anything, please go ahead. Super. So Nathan, I think, is on the call. I think, Nathan, you're going to do the first update.
That would be fine. Yeah. Thank you, Paul. I guess if you stop sharing, then I should be able to share. Looks like the participant screen sharing has been disabled. So who's ever hosting might need to enable that.
Okay, let me see if I can find it. Can you not share, Nathan?
When I try to share, it says the host has disabled participant screen sharing.
Oh. Right, give me a moment. Sorry, I thought I'd enabled it. I can't see where it is. So share screen. Advanced. I'm really sorry, Nathan. I can't see where to.
That's okay. I don't know if under participants, if there's like a drop down on a per participant and you enable a specific person to share.
Okay, should be okay now. So I've put all participants.
All right. Looks like I can do it. Thank you very much.
Okay.
Great. Well, thank you very much to Karen and Paul for organizing this. Fantastic to see and speak to people if only virtually. I'll just be sharing this one single image from the United States, the Department of Energy's Agile Biofoundry. Just as a reminder, we are a distributed biofoundry, so we are not operating in one single location in the US. With our current Department of Energy and the current US administration, kind of our top two priority areas are decarbonizing industry and pursuing sustainable aviation fuels. Our main goal is still to reduce the time to commercialization for bioprocesses. And we are still doing that through our integrated design, build, test, learn cycle, along with things like techno-economic analysis, life cycle analyses, host onboarding, and process scale up. Very importantly, we're still focusing on collaborating with both industry and academia, and I'll have a couple of updates on that. And then a really kind of a new and revamped effort is really trying to promote diversity in science, technology, engineering, and maths, and workforce development. I'll have a couple of quick little updates on that too. So at an overall level, our foundry is healthy. In the US, our Congress has at least have the language for increasing our budget from 20 million US to 30 million US per year. They haven't passed the budget yet, so that should be happening in the coming weeks, with an increased emphasis on collaborations as well as on machine learning and artificial intelligence. In terms of some of these collaborations, a really exciting thing that happened to us over the past months is that there's been increased coordination between the National Science Foundation in the US and the Department of Energy. And the National Science Foundation is going to be funding the academic side of the collaboration, and the Department of Energy will be funding us. A number of you actually participated a couple of years ago now in a workshop that was trying to figure out how we could do this better, and it's actually borne fruit. So now we're actually getting to the point where the academics will be submitting proposals soon. So that's a pretty exciting development. Other things that are kind of interesting in collaborations. We are currently having a visiting postdoc from VTT in Finland, and we're going to be kind of cross-comparing methods for working with the fungal organism Aspergillus. So looking at the automation of transformation, kind of bioprocess scale up. So it's very exciting to be, working in a project that kind of came out of the Global Biofoundry Alliance with UC at VTT and the postdoc Mati. So that's going to be really fun. Also kind of along the promoting STEM, we are setting aside $1 million in the coming year to be working together with minority supporting research and development consortia members. So that's going to be a very interesting new kind of collaboration area for us. And we're also starting to do things like, for example, for some of our web applications, we're working to make them more accessible for the visually impaired. So we're learning more about how to make some of our tools even more accessible. So again, thanks to Paul and to Karen and for everybody. But maybe I'll leave it there for now. Thanks.
Super, Nathan. Sounds really exciting with the NSF getting involved and developing projects. That's a really super development. That's really cool. Great. If anyone wants to ask a quick question, please go ahead. Or we can shoot forward, whatever people want to do. Great. Okay.
Yeah. Question, Nathan. You just mentioned the annual fee is about $20 million US, 20 to 30 million US dollars. Is from the government or from the industry?
Yeah, thanks for that question. Those money numbers right now are coming purely from the government, from the Department of Energy. Above and beyond that, industry can do fee for service, or they can themselves pay out of pocket for collaboration types of work. But the money numbers that I just gave you there are purely from the government.
So the purpose of the foundry is to serve both academia and the industry. So how to balance the services provided by the foundry to both quite different communities?
So in terms of our collaboration projects, we have, if you would like to call it, different pots of money. So we have specific money set aside to work together, for example, with NSF funded academics or those from the minority supporting research and development consortia members. And then we have separate other resources set aside that are more likely to be for work with industry. So there's different funding support for those two. And I think in terms of our technologies or our host organisms, our capabilities, I think they're fairly broadly applicable. So maybe the questions that are getting asked might be slightly differently, but hopefully we're applicable across academia and industry.
So, yeah, thank you for the wonderful answers. And I'm still because in Shenzhen we are now establishing the Biofoundry. The purpose of the Biofoundry to serve. 熊飞，你把你的那个 mute 掉。对。So, somebody is speaking. 熊飞，你把你自己的 mute 掉。Oh, sorry.
Yeah,
so fine. Yeah. So the question is to build a foundry. to serve both communities, because the purpose from both communities are quite different. From the academia is in most cases is from the individual laboratories. So it's small scale and is very the growth is really diverse. And for the industry is a very fixed goal, and for a certain company, they probably have a very defined purpose, and I would say the contract-based research, it required the foundry to have a team of expertise to support this industrial purpose, which cannot be developed easily in startups. And on the other hand, for the academia laboratories, sometimes they have expertise to use the foundry by themselves. So how to balance these two quite different purposes in a single foundry is kind of we look at as challenges for I would say the versatile biofoundry.
I mean, if I can step in, I think it's a great question. And yeah, go ahead. Sorry, Nathan, go ahead.
I was going to say, yeah, it is a great question. It's probably one that a lot of us are struggling with. We're not trying to be everything to everyone. So all of our collaborations generally will go through some type of a competitive process. If it is with government support, if they're just paying out of pocket, then they can kind of decide if we're the right fit for their needs. But a big part of the competitive selection process is making sure that we actually have the capabilities that are well suited for that particular group's needs. And also that there's merit behind what they're doing, and it's going to be a good use of our resources. But it is a big challenge, and there's only going to be maybe certain interactions that are going to work well for both sides. But we can talk about that more later, maybe. Yeah.
Yeah, it's a great question. I just want, let's move on, but let's keep that question alive, because I think it's a really important one, and certainly there are different models for how one does it, different scales actually as well. If we can move on, I'm not sure, is Claudia on the call? Is she talking? Because I know she's accepted a new job just recently. So I'm not sure whether maybe it's Tom Williams. Yeah.
I'll just say hi. It's Janet here from Brisbane.
Oh, Janet.
Hello.
Okay.
Claudia, so we've got massive floods here in Queensland at the moment. So it's entirely possible that Claudia just can't make it for whatever flood-related reason. Yeah, there's loads of people without power, and anyway.
Oh, goodness.
Don't know.
Gosh. Okay.
Yeah.
Thanks, Janet. Okay, no problem. Well, let's move on. I think we've got Tom Williams, maybe, or that could be a similar problem. I don't know. Oh, Tom, you're there.
No.
Tom,
we've got quite a bit of rain, but not quite as much flooding as Brisbane, but we're good.
Great. Okay, so Tom's going to tell us about the Australian Genome Foundry. Great. Over to you, Tom.
Thanks, Paul. I've kind of been observing this Biofoundry Alliance from afar, but haven't participated before, so it's nice to be here. I'm representing the Australian Genome Foundry. We're based in Sydney, Australia. We don't quite have views like this. We're a little bit away from the water, but we have just refurbished a really nice new lab to house our Biofoundry. So we're at a pretty early stage of development, in that for a long time we've been accumulating equipment and using the Biofoundry basically for in-house academic research. We've had funding from a variety of sources, and we're predicting to hire staff and properly begin operations later this year. In terms of where we came from and what we do, we've got a bit of a history with the Yeast 2.0 project, aimed at engineering a synthetic yeast genome. And at Macquarie University, we've been building two of yeast 16 chromosomes, 14 and number 16. And we kind of use this project to establish some basic automation protocols for high throughput real-time PCR and DNA extraction. And that kind of springboarded us into looking to expand our Biofoundry under the leadership of Starkey Pretorius and Ian Paulson. And we wanted, one of the goals that we have, or the visions that we have for our Biofoundry, is to maintain this capacity for very large scale DNA synthesis, synthetic genomics, and that type of research. In addition to the basic capacity of large scale DNA construction, we're pretty interested in novel forms of genetic diversity. So that's pretty relevant to the Yeast 2.0 project, which has the scramble system, which I'm sure most of you are familiar with, inherent to it, which can shuffle chromosomes and change their architecture and gene content in semi-random ways. In addition to that basic capability, we want to set up I guess the full stack, I suppose, of what all of our Biofoundries are probably aiming for, where we can screen very high levels of fully random or semi-random genetic diversity using growth-coupled approaches or biosensor approaches. Inputs to this could be scramble, random mutations, novel protein fusions, things like that. And we want to span this entire, I guess, funnel of screening right down to small numbers of strains in deep well plates and eventually bioreactors. We do aim to, I guess, specialize at the top end of this funnel. And for detailed omics, we'll look to collaborate with others, in particular, Lars Nielsen's lab. In terms of our overall strategy, I'm interested in the input of others with this, but the basic idea that we have is that we want to service academics, other scientists, as well as big and small companies. In particular, I think we have a focus on incubating very early stage synthetic biology companies, with the idea that they pay a small access fee and they do everything themselves in the foundry. This has suited us quite well in our early days where we don't have staff to run operations just yet. And we already have a company incubating with us at the moment. The last thing I want to touch on is the ARC Centre of Excellence in Synthetic Biology. So the Australian government in 2020 invested quite heavily through this particular grant, which is worth about $50 million and spread over, I think, 19 different chief investigators in Australia. It's an academic grant aimed at doing almost sci-fi things, which are becoming now mainstream, such as microbial communities, synthetic organelles, and what we're calling neo-biochemistry. So we're planning for a lot of interaction with this consortium through the Biofoundry to, I guess, test fundamental hypotheses based on these topics, but also to serve local industry and incubate very fresh companies. The Centre of Excellence is still in its early days. It's funded for 7 years. If anyone has interest in these three main themes, we are open to seeding new collaborations through that as well. And yeah, that's all I have for today. Thanks, Paul.
Thanks, Tom. We're okay for 1 or 2 questions if anyone has anything specific.
Just one quick question. Is Biofoundry part of the ARC Center?
No, it's not directly funded. Yeah,
Oh,
so
okay.
it's a separate pot of funding. Yeah.
Great. I think this model that you illustrated between companies and academia, it seems to be a very, very common theme for many different foundries, and it'd be very interesting to compare notes on what works and what doesn't on that one, maybe later on.
Yeah, absolutely.
In our discussion. Great. Okay. No more questions. Then we can quickly move on to hopefully if Stephen Hallam's on the call. Maybe not. Okay. If Stephen's not on the call, we can, if he joins later, we can slot that in. And we have, so moving down the agenda now, we're ahead of our time. But anyway, we have Panwan Binay, are you around? Oh, yes, there you are. I can see Panwan. Okay, I'll pass it over to you now on Biofoundry India, and we can come back to Stephen if he joins later.
All right, I'll just share the screen. Can you guys see that?
Yeah.
Yes.
Okay, good. Thank you very much. And thanks to Paul and Karen for the invitation. We are probably one of the newest members of the GBA, and you know, we had hello.
Binay, sorry, I think probably you need to do it on the full screen mode.
Okay.
You're in the presentation console. Now it's fine.
Okay, good. So thank you very much, Pawan. We are probably one of the newest GBA member, and we had a very bad, devastating Delta wave. And in fact, the idea was to start last year, but we couldn't because the university was completely closed. And I'm joined by Professor Pawan Dhar. So both of us are professors in School of Biotechnology in one of the largest public universities in India called Jawaharlal Nehru University, located in New Delhi. And we are late because of, as I mentioned, both the Delta and the Omicron wave, but the Omicron was not as bad as Delta in terms of the life loss. So we are planning to have two focus areas in our Biofoundry. The first one, which is going to be headed by Professor Dhar, is the dark matter of the genome, essentially harnessing the non-expressing part of the genomes from various organisms sequenced so far, and some of the sequencing that we will be doing in-house for India-centric organisms. And the second one is actually harnessing the power of software that India already does for the rest of the world, and also coming up with processes for both the different parts of the Biofoundry, including the AI. And the challenges that we have here is probably the same as many of you do face in your geographies. The first of all, we don't really have a successful use case of running large shared facilities like Biofoundry. And primarily the reason is we can't be competitive compared to the private players out there in providing services, because most of the private players do negotiate bulk discount of reagents from companies, and we can't really do that because we have to guarantee a certain number of volume in terms of dollar value to do that, and we are not in a position to do that. The second and the most important part of our challenge is that our government actually does not provide long-term core funding for large resources like Biofoundry, particularly for personnel. So in order to hire a lot of people to run the core facility, we will have to do temporary staffing, and it's very difficult to get at a higher level, particularly independent scientist level, to get those personnel costs covered from our grants. So we can cover it maybe for 2 to 3 years at max, and then it goes for the renewal. But most of the larger facilities like Biofoundries are never renewed beyond the second term. So it's very difficult to get that. We really think that we need a solution there, and we'll be more than happy to listen to some of the suggestions that many of you probably have already faced this. And we had many discussions with Paul and others. And we think that we can use our Biofoundry for large-scale COVID testing. And I think this is one of the model that, for example, Paul had done in the Biofoundry there. And if we do that, then there is funding, provided that COVID is here going to stay, because if it doesn't stay, then we have the same problem of funding. We do think that we can provide services in certain areas that the private players do not provide, and both in terms of the Biofoundry-based experimental services, but also consulting services. Unfortunately, in India right now, government universities, public universities do allow professors to be consultants for a part of their time, although very restricted, but it may be very strange for you guys, but we really work on a socialist system. So we think if the government, the public is paying for our salary, we should not be really charging any money, or we should not be doing any of the jobs outside our normal teaching and research job. But fortunately, our government has now allowed that for a percentage of the time. And third, but not the least, obviously, that we think there could be some global funding where we can tap into as a GBA member in collaboration with other partners present here. And everything that we plan to do, both in the areas of non-expressing part of the genome and also in software and AI, is actually published. So any of you want to know on these ideas, obviously we haven't really done much except in our individual labs. in the synthetic biology in the recent volume. So with that, I'll end, and love to learn more from the other members, or if you have any suggestions or comments, love to get that. Thank you.
Thank you very much, Pawan. I think someone in the chat wrote, I think you're in good company in terms of the challenges. I think Nathan mentioned something. But I think the floor is open for any questions or comments or advice.
Just one quick question. Are you in discussion with any other universities in India?
You mean in India or outside of India?
Yes, in India.
So the idea of Biofoundry India is to work in a hub and spoke model. So in the future, what we want to do is that we want to create the hub in our university in Delhi, but also have different spokes around. But it's very easy to build a model, what I just told, but it's impossible to sustain a model. So I think we really do not want to expand our model unless we see sustained funding. And I underline the term sustenance, not the funding at one-time affair. So I think if we get personnel cost covered for at least 2 or 3 people in a longer term to hire at a scientist level or even staff scientist level, then that is what we are looking at. Because if we have different members around the country, but they don't function beyond 2 to 3 years, then the purpose is not met.
I'd just like to say I'm looking forward to seeing what comes out of your software development. I think that's something that's probably top of mind for a lot of people as well.
One of the things probably we can discuss during the discussion time, Paul and others, is we have different, what we call resources or the strengths. Different people at different geographies have different strengths. So I think GBA as a community, if we can pull our strengths together to solve one large unmet need, and because in India we can do things in software with other people can't do at a price point. So we can have volume at a price point which is impossible to match. So this is something that we think we are in a unique position to offer to the community, but there has to be one large scientific projects where most of the GBA members are interested in, because otherwise it doesn't really work. But if most of the people are interested in a large scientific problem and different people pitch in based on their strength, I think to me, that's a doable model.
Thanks, Vinay. I think we'll definitely come back to this in the discussion point later on. Great. Well, thank you so much. Really good to hear what you're doing and what you hope to do. So moving on, if Vince is on the call, I'm not sure. Vince on the call? No. Okay. Janet is definitely on the call, because you've said if you. So Janet, we'll hand over to you, and if Vince or other colleagues join later, then we can slot them in.
Hi, Janet Reed. I'm from the CSIRO Biofoundry. Sorry, that's CSIRO. We just call it CSIRO here, so I sometimes will just slur the word like an Australian. Just get used to it. We're based in Brisbane, Queensland. We were a joint facility between the University of Queensland and CSIRO, and brainchild of Claudia Vickers, a bunch of people at UQ and a bunch of people at CSIRO. And now we're solely funded by CSIRO. So I'm decided today to focus on, just going to share. I'm going to focus on a couple of the things that are of key interest to the Biofoundry Alliance through the working groups. So I've picked out 4. How's that? Looking good?
Not yet.
Sharing?
It's coming. Not yet. Now.
Hopefully in a second. Yay. Okay, great.
Yep.
Okay. Yeah, so we're a small biofoundry. We've only been running for about 3 years now. So we've got lots of fancy equipment, you know, all the usual things that people have, like few liquid handlers, echo robots, all that kind of stuff. And we've been focusing mostly on the build stage of the design, build, test, learn pipeline. And we're also relying on CSIRO's extended network, because CSIRO is Australia's national science agency. There's a network of facilities and sites across Australia, all specializing in different fields. And so through that, we can access strain engineering, as well as extended testing facilities. There's a bit of scale up into fermentation, and we've got access to Data 61, which is an arm of CSIRO, which and they do a lot of machine learning and AI development. So we're kind of, while we have this discrete package of work that we try and focus on, we do try and tap into CSIRO's network of expertise as much as possible. And that's something that I've been trying to develop a lot over the past couple of years that we've embedded in this new facility. So one of the things that I'll start with is the business model, sustainability and value proposition. So that's one of the working groups. So we had an independent consultant come in to look at our business model and you know how it would fit in with CSIRO's mandates. We work in an operating environment where we can't compete directly, undercut any Australian businesses. So we have to set our fees as a service at the market rate, which can be quite difficult for academic researchers to access, and that's something that we've found is a real constraint on our ability to work with academia at the moment. There are ways around that through collaborations, but yeah, it's been slow going to try and build that sort of. There's a sort of like a little impasse here where CSIRO generally will work as a more sort of like focused on industry, service to industry. So, and they see us as a service to industry. So having that change in focus to allow us to move more into an academic space is a little bit constraining at the moment. But as I mentioned, there are benefits to being in CSIRO, which is the extended network of services that we can offer as part of a package to a potential client. So we also don't think that from the business model, and this is clear across all the biofoundries, that actual sustainability is an issue for everyone. Being able to raise enough funds to hire enough staff and keep equipment up to date and running is a big ask. So, and that's been recognized by CSIRO that we probably will not be a fully sustainable, self-sustainable entity. So the focus is now on the value add to bioeconomy development for Australia. So that's sort of like working with industry as much as possible to develop the Australian bioeconomy through synthetic biology. The next one that I'll mention is our work in the sort of data quality, reproducibility of experiment types, metrology. That's one of the working groups. So for us, we're really focused at the moment on process development because we're new in the space. Every project that comes to us is different. We work up every project from scratch. We are not at the point where we can say that is a routine process yet. I mean, there are some, you know, plasmid prep, yeah, of course, but you know, we're trying to move to a more, you know, more streamlined processes through the Biofoundry so that we can sort of like say, this is a package of work, this is the way it will work. And I'm sure it's the same for everyone else, that you find that there are, you know, when you're in the earlier stages, there is so much variation between projects that, you know, you find that you're working up everything from scratch, and it can take a lot of time, a lot of effort, and we're working towards getting those solid processes in place. One of the success stories in this has been the machine learning project development, which has been run by Maciej Kolko, who many of you will know from the GBA. He's been working on a couple of machine learning projects with Data 61, and we found that we have got to a stage where we're getting reproducible iterative cycles in these projects, so running through 5 and 6 cycles and having reproducible data. So it's wonderful to see something that is kind of like running over a long period of time that is giving us sets of data that are useful. Moving on to software. So there's a software group, but I stuck hardware on the end because, you know, to me they fit together. We have Teselagen LIMS system. So for our design and build stages of the pipeline, and we're trying to work how we're going to do the data management side on how do we integrate the data management on that as well. So we're going through a lot of process at the moment of working with what comes out of instrument, the type of file that comes out of instrument, doing the data conversion that is necessary for upload into Teselagen, you know, and then download back into the next instrument, and then just that management of data, the pipeline of data that comes as you transition through the stages. So we've got a data engineer on board now, Adrian Marsh, and so it's been really fantastic to be able to have that capability. And he's also working at integrating in ThreatSec, which is for sequence of concern screening, and that's necessary because we're, you know, quite facing industry. We're working with, we're not working with just academics, we're working with a wide range of people. We need to have robust processes in place that we can say, well, what the product that we're producing has been screened, and we know what we're delivering to you. So he's developing the API integration into Teselagen and working through that process at the moment, which is great. So the last part of this is the hardware, and so we're doing a lot of capital expenditure predictions for the next few years, working out what sort of equipment that we need. We kind of bought you know, the base core instruments that you might think you'd need for just the molecular assembly work that we do, and now we're thinking about, you know, what sort of added capability do we need to complement what's available in CSIRO, or make something that's in CSIRO at the moment a little bit more high throughput. So they, you know, they might need, you know, something that's that a little bit that can sit in the Biofoundry, works well with what we do to continue that pipeline. So one of the capital expenditure items that we purchased this year was the side effects SRT flow sorter, which can sort into plate, and I love it, just love it. Yep. So very happy with that. But there's lots of other things that we're thinking about, and then we start thinking about how we're going to integrate all those instruments into work cells, and to increase the amount of automation in the laboratory, so we can reduce the amount of hands-on by the technicians, and they can start focusing on managing the data and the experiment planning more than just the moving a plate from one machine to another. So I'll leave that one there. The last focus will be on outreach, industry policy, etc. So this one's a bit of a mishmash. So maybe I'll start at the bottom. So we used to be funded by the SynBio Future Science platform, which is an arm of CSIRO Biofoundry. They're actually coming to a close at the end of this financial year in June. And they've been a great support to us. And many of you know Claudia Vickers, but also Alison Rice and Colin Scott, who are also members of the Future Science platform. That sort of soft cushion that we've had for the past 3 years to get us going, we're now moving into CSIRO wild waters, and so we're moving into a different space where we'll need to become a little bit more competitive with the way we manage things. So we're going through this period now where we're trying to solidify things so that we are much more competitive and that we can work well with the CSIRO model. We have good levels of state and federal government engagement, and that's all through the SynBio Future Science platform. So all tribute to Alison and Claudia for a lot of work that they've been doing in that space for us. So that comes to you know, talking about the Queensland bioeconomy, as well as engagement with the federal government's most recent Trailblazer funding program, which should be seeing some funding going through the universities to access CSIRO platforms. So we're looking to see which universities will engage with on that. So that would be really fantastic. And we've now got a website finally, yay. It took a while to get us there, but you know, we have something that we can now present to the world. And we're also working with the Australian, I guess you call it Defence Export Control, to work with, to seek advice and also give advice on how we manage our synthetic biology output in terms of exporting to overseas partners. So that's been interesting as well. And I'll leave it there, and I'll just say a big thank you to Claudia Vickers for the time that she served with us. She's been a driving force for getting the Biofoundry going. So thank you.
Thanks, Janet, so much. That was great. Superb progress, really interesting to see. Lovely, lovely talk. Yeah, we're open for any quick questions or anything. There's some comments in the chat.
Hello, Paul.
Yeah.
Hi.
Yeah.
This is Renus
Yeah.
from Edinburgh. Very nice talk. I just have a quick question with regards to your threat screening. Have you received an updated database? Because we are screening as well, and the database we have from IGSC, it's since 2018, so it hasn't been updated. It's one of the monthly discussions we are having. So I was wondering if you have updated your database there.
Yeah. So ThreatSec do all that database management at their end. So we don't have to receive. So we basically, they've got either a web service or we can access through the API. So I'm loving it, because bit black box, send sequences, comes back with a threat rating for each of the sequences, and we can we're looking at ways that we can automate that, looking through those sequences, so we don't need to use a human eye necessarily to go through that. Yeah, but there are other companies out there that are now starting up in that space. There's another group called ACLID Bio, so that's ACLID Bio, and they may have approached a few of you recently, but yeah, they're also doing something very similar to ThreatSec, but they'll also do a lot more data processing as well at the other end, bioinformatics on it. So you can not just sending you the results, they'll actually, you know, go through it for you as well. So the level of support they can provide is different. And I'm trying to build the cost of this screening into what we charge to the client as well. So you know, it's scary when you look at the price and you go, oh my gosh, but you just build it in like you build in the cost of the DNA bases that you order from wherever. And we only screen, we don't have to screen everything as well. We can screen the design, the final designs. We don't have to screen the individual parts that come through from IDT or whoever, because they screen for us, which is fantastic. But, you know, in terms of any material that the client sends to us directly, we'd need to screen that. So if they send us a strain, we can do that. Yeah. Sorry, I'm babbling now. I'll stop.
No, that's really helpful, and it's a kind of a very interesting question as well. And maybe we can share some ideas on, or you know, any practical aspects of that would be helpful for sharing to everyone to know what tools are available, what people are actually doing. It's a very key area, I think. Great. Okay, I'm conscious of time. Thanks so much, Janet. That was great. And I've got to move on to Lars, who I think I saw was on the call. Lars Nielsen from, yeah.
Yeah, can you see it? You can see it. That's good.
Very good, Lars.
Let me go ahead. So basically, I'll give the Biosustain, which is the left-hand side there. So sort of splitting 50-50. I'm actually going to Denmark on Sunday. So finally, I'll get over after 2 years. But the bottom part there is the CFB Biofoundry or Biosustain Biofoundry, but we also have some partnership, as Janet referred to, in the Bioplatforms Australia. So I'll quickly cover that. So when we established CFB 2.0, so the extension of this center of biosustainability, we really built it around the idea that data generation and data analysis is critical for future science. So we allocated quite a lot of our resources, close to 70 people, into broadly what is a biofoundry. Now, that includes basic research in new algorithms by Bernard's group in San Diego. It includes some basic build and test at CFB, but it also includes a very big data integration platform, because that's really the focus. And then, of course, we have some application areas in sustainable chemicals, products, and microbial foods, which is sort of our internal use of the facility. We are not we don't reach out except for very specific partnerships that fits into our research model. So that's a nice thing. We have it all funded without the external funding. If we look at the build and test, we very much build on engineering on the genome. So ALE to introduce mutation, library generation using Onyx of mutations, and then rational engineering using CRISPR to implement those candidates, or get strains from the outside world. We have a test platform for cultivation and extraction, and a very strong focus on deep characterization, so full omics characterization. And this can be getting 4,000 strains from Novozymes to sequence to build up a pan-genomic database. It could be our 100,000 ALE variants to understand and characterize those, or it could be much more targeted engineering a strain, looking at its phenotype, and then do deep RNA-seq and MS-omics characterization. So on top of that, we have some research project. Genome engineering is really what it says, actually building genomes, not building parts and putting into genomes, but building genomes from scratch. So that's really next generation of foundries. And transporterome is really how to use the platform to screen through and identify the function of all the transporters that are annotated as Y proteins in E. coli, etc., because they're important for metabolic engineering. And then we have some house services that we also have to address for the institute as a whole. It's 300 people in the institute, so there's quite a bit of work just in that. And what we have that is quite unique is these 22 people really focusing on how do we take the data we generate and contextualize it. And it's built very strongly around network machine learning, so Neo 4G type, 4J type framework to take all the data that is in the literature, everything we can find in NCBI, bring in our own PDF files, and then contextualize what we have observed in our data in that case. We will be opening this up. We're just making it stable enough that it can run. It is an open platform, so we will make it available, but let us get it stable first. Shifting to what we're doing here in Australia, so we're sort of working more on secondary screening and how do we use deep characterization to help design in a normal biofoundry, and really focusing on building up deeper data set and taking it the next step to scale up. Now, Janet already mentioned we have a new framework here for some very large industry driven project or university industry partnership. that are drawing also on CSIO. So in that case, the CSIO Biofoundry would be the natural partner in this company with the idea side, and we would help with some of the deep characterization of those strains. I think it was worthwhile sharing some thoughts I had from Darren. This is back in 2017. I think we had these deep discussion of what the Biofoundry is. So he was responsible for setting up all the informatics for Amaris. So, and he's now at Demetrix. But basically, this idea of a Biofoundry generating a lot of complex data and drawing on a lot of complex data, and it makes it basically a computer peripheral. I thought it was clever when I said that, but basically somebody had said it well before me. Mirwald from Microsoft had made that observation before. And the other thing is this idea that we don't actually accelerate strain build, but we escalate. So we build 100 strains instead of one. And from a design point of view, that actually matters, because suddenly you have to make 100 informative designs unless you just want to boil a lot or burn a lot of money. That's one of our challenges, that it's hard to make good designs, and that's why we have some focus on model-based design. So this is just this emphasis that everything we do either draws on data or contributes to data, and then data integration is critical. Most of the strains will actually disappear from physical embodiment and end up just being data, and it's how do we use that information as we go forward. So we work with Amaris on setting up a project on using deep learning or deep characterization with omics on a couple of their projects. Now this was with Darren and Joel, and now both of them have gone to Wheat, or at least both of them work on CBD projects with two different companies, so they left Amaris. But fortunately, Kirsten and Sunil are still very supportive of this, and we just finished this project where we took a couple of projects at early stage and said, can we use omics to do deeper characterization? So this is classical fermentation, except many fermentation in parallel, proper fed batch with supernatant and extract of intercellular metabolites and proteins. And we have nice gas analysis, which we use to control our fed batch process. Downstream of that, we have automated our sample processing. So we have quite a bit of equipment around extraction and processing, and quite a bit of analytics to do the deep characterization. So it's really about generating large data sets. And in our experience, the OpenBIS framework has actually worked very well for integrating all these data from fermenters from all our machines. So they pull the data in a reasonable format. So that's what we are using. We can use all the machine learning data mining things, but I have a very particular strong interest in kinetic modeling. So we tend to set up kinetic models. So we will take a framework, set up a detailed model of the system. We will extract everything we know about the enzyme, characterize them well, and then we can use a single data point to start generating kinetic models that could describe this particular observation. In this case, it was wild type Putida, but we wanted to increase the amount of acetyl-CoA in order to make PHP. And we did a classical metabolic control analysis, identified a couple of genes, and then by manipulating those genes, we were able to push the titer or double the titer inside the cells. Now, with the Amaris project, can't talk about what exactly it was, but basically we were able to push it almost up to 10-fold higher, and they tested that in large-scale bioreactor, and it actually works. So the basic idea is there for it working. So that was it for today.
Thank you very much, Lars. That was a great update. And Giovanni put his hand up, I think.
Yeah, great talk, thanks. Quick question, how many customers come to you because of the design skills that you have put together?
So on our Australian side, we have been doing this for 15 years. The only thing that has changed is number of strains. So 15 years ago, we worked with Dow on one of their projects. They gave us 4 strains, and we characterized 4 strains in great depth, come up with a good target, and that fortunately improved. So we have companies like Lancetech, they are in the other end, they want to characterize a couple of thousand strains with us. So we have quite a lot of companies working with us to trying to find the next non-obvious target.
I loved your quotes, by the way. Escalate and computer peripherals. Great, I'll remember those.
Well, I think it helped us a little bit in thinking about the problem we have, which is very often.
I was going to ask you very quickly, and I know we need to move on. It's wonderful developments in DTU. I mean, when you say it's going to be open, how do you envisage a sort of community-driven contributions to this project? Yeah.
In this case, it was a data platform we will open up. I am not convinced about this service. A lot of different customers. In our hands, it takes 6 to 9 months for every single process we want to develop for every single strain.
Right.
So we will specialize in a few things, and basically, well, if you like it black, we'll make it. If you like any other color, we're not going to. And I mean, we spent 10 years in CFB 1.0 trying to get robots to work and doing everything for everybody, and nothing ever worked. Basically, I threw the machines out, I threw the people out. I basically got a whole new team for the build side, a whole new team for the analytical side, and said, we are doing these 5 things and we do them well. And I think that's a different strategy, but I think all of us have to specialize rather than promising everything for everybody. I think that's too hard.
Very good.
There's enough of us that I would rather see us send it to people who know what they're doing rather than all of us trying to do it for everybody. But that's
absolutely. That's a very, very good point, Lars, and one perhaps we can discuss later. Thank you so much. Moving on, if Nicola's on the call. Nicola's there. Great.
Let me share my screen.
Sorry, dear.
That's coming through. Oops.
Perfect.
Right. So I will briefly introduce the Erlen Biofoundry. So for those of you that don't know, we're based in the Erlen Institute, which is an independent life science institute on the Norwich Research Park, based over in the east of England. So this research park houses a university hospital, several research institutes, and an innovation centre that hosts around 50 or so small businesses. So I guess it's important to note that our Biofoundry sits in an ecosystem of interlinked facilities across the park, which includes genomics pipelines for next generation sequencing. We have a single cell and FACS analysis platform, bioimaging platforms, metabolomics platforms, and biophysics platforms. So those things that don't sit within our physical Biofoundry lab, we have close access to, and those facilities are used to interlinking with each other to bring larger projects together. So we were established in 2015 with a £2 million capital equipment grant from our National Bioscience Funder, which is called the BBSRC. And since 2017, our operations have been funded through a national capability grant, which supports us as a national capability in engineering biology. And this enables us then to employ a Biofoundry manager, automation specialist, and have some operational funds. So this is a 5-year grant that's been extended to 6 years due to the pandemic, and we're currently preparing a grant submission for the next 5-year period. And the goals are generally then to provide researchers in academia and industry with access to equipment and expertise required to undertake large-scale experimentation and to, drive the bioeconomy, particularly to complement the strengths of the research park, and to train researchers in synthetic biology approaches and the use of laboratory automation. So this is a simplified version of our operational model. So we interact with scientists and in industry and academia through 3 routes. And the main route of these is through the progression of collaborative projects. And these projects then produce a number of automated workflows that we make available to others as services, kind of smaller services. And those collaborations then often involve hosting researchers within the Biofoundry for anything from a few weeks to several months. for whom we develop training resources, which we're then also making available as short, either in person or virtual training resources. So our collaborative projects then are either funded by grants, in which the Biofoundry is a collaborative partner, or in some cases, particularly where we want to kickstart collaborations, users will fund all the consumables and equipment use, and we'll kick in our expertise and time, usually aiming to build at least sufficient preliminary data to put into a grant application. And then access to our service workflows are usually provided under a full cost recovery method, and training we just provide to the community more or less for free. So to give you an example of the kind of projects we've been doing in the last few years, one of these has been automated growth challenge assays. So this is looking particularly to identify the genetic basis of how one bacteria may inhibit growth of another. So looking for the genetic basis of new antimicrobials. So we have some pipelines that we've developed here, which include library production and arraying and quantitative phenotyping. We've also, so my lab's plant synthetic biology research. So we've been working a lot with plant enzymes, particularly for reconstructing metabolic pathways. So we've developed a nanoscale expression and characterization pipeline, which involves build, cell-free protein synthesis, and enzyme assays and targeted metabolomics. We've also been working on a project with Colorifics, who are a synthetic biology company producing dyes for textiles. This is an example of one of our grant-funded collaboration, and here we have a mixture of builds, microfermentation complemented by microfluidics, and going on for molecular phenotypes. So that's it. There's our contact details. We have a new Biofoundry manager starting next month, so we have had a bit of staff turnover. We particularly tend to lose our automation staff into industry, which is disruptive for us, but also I think speaks to the growth of this industry and the importance of how we're viewed as a facility as well for training and providing an expert workforce into the UK bioeconomy. That's all I got, Paul. Thanks.
Well, that's super. Thank you so much, Nicola. That's a great update. Any questions for Nicola? I mean, I'm beginning to get a lot of common themes emerging from all of these talks, which is very interesting, and we can talk about that towards the end. Great. Okay. The next talk is from Akihiko Kondo, I believe, if he's on the call.
Yes.
Ah, hello.
Yeah, hello.
Very nice to see you after all this time.
Very nice to see you. But I'm sorry to say I didn't realize that I should give a short talk, but I tried. I prepared something. Yes. Can you see?
Full screen mode. If you put in full screen mode, perfect. Great.
Yeah. Can you see?
Perfect. Wonderful.
Yes. So sorry, as I mentioned, that I didn't prepare so well, but I tried to explain you what now we are working with. So as a university Biofoundry, we are actually developing the many tools and methodology. So in a sense, so we are working with several aspects of this Biofoundry development. So as a design, we're still working for the improvement of the design tools. So I don't go into the detail, but we have developed many different type of the design tools. And recently, for the build part, we are putting some effort on the construction of the several parts. For instance, still we need a lot of many parts like inducible promoters and so on. Those kind of variety of the new biological tools, actually parts we would like to have more. So in a sense, we are putting some effort to quickly develop these kind of the genetic parts. So I just would like to show you the one example I just recently published this results last year. So this is inducible promoter for the yeast. Especially in this case, we are working with the Saccharomyces cerevisiae and Pichia pastoris. So we have developed several very nice inducible promoter system for the yeast systems. So this is actually trifunctional selector system to quickly develop this kind of genetic tools. Actually, this is an example. This is, for instance, inducers, and actually this is, performance of the promoter. So something like this. We have developed a variety of the, genetic parts. So we are putting a lot of effort on that. And another thing is we are working with the quick construction of the artificial enzymes, because, it's quite important to develop the artificial enzyme to construct the artificial metabolic pathway. So in a sense, I think, we are putting a lot of effort to develop these kind of the variety of the, this is not so special, but anyway, we are working with pretty much with the automation for the development of the artificial enzymes. And the test part, this is rather special for us because fortunately in Japan, we have a Shimazu company. We are very close to the Shimazu, so we are pretty much working with the Shimazu. And this machine is, sorry, this machine is an automated pre-treatment system for the metabolomic analysis. So this machine can carry out 300 samples of the pre-treatment per day for the metabolomic analysis. Because this is robotic systems, the accuracy of the metabolomic analysis improved quite a lot. So and then we are working together with the Shimazu company. This is called the autonomous lab. So this is automated analysis by using the mass spec from the colony. So in a sense, all the process, because in many cases we need a mass spec analysis and so on for the screening. So in a sense, we together with the Shimazu company, we are trying to develop this kind of the fully automated system. We call this autonomous lab systems. So in that sense, our foundry working quite closely with the Shimazu company to develop these kind of the new types of the automated machines. And also we are working with for the test part, we are working with this kind of the microbiome reactors, because if you try to develop some new molecule which control the microbiome, we need this type of high throughput microbiome systems. So in essence, this is a rather big one, but also we are working with the multi-plate. So like this, we have working quite a lot with the microbiome, in vitro microbiome analysis systems. So this is important. I think it's important for the test part. So like this, this is just prototype of the Biofoundry. This is for the education of the company people, and of course we can do the work. We can work with the company. So we are currently working with 20, 30 companies. Some so in a sense, a lot of more and more customers come to the university to work with this kind of very small types of the prototype of the Biofoundries. Because at the beginning, company cannot invest so much. So in a sense, at the beginning, our university people are working with this kind of prototype Biofoundry system. It's not so high speed, but it's a kind of a good opportunity for the company people to for the training and to work with at a rather low cost. So, but that is not enough. So in a sense, we have started our startup company, this company called Bacchus Bioinnovation. Probably you understand this Bacchus, Bacchus is a Greece god, The reason why I named this company Bacchus is I love red wine. So in a sense, this Biofoundry company is Bacchus Bioinnovations, So actually this is a lab of the Bacchus Bioinnovations, So I just started this company last year. So so far investment is not that big, something like $50 million US, and as just a Series A. And as a next step this year, we would like to have 10 to 30 million US dollar to the scale up. So like this, because the reason why we started this kind of company is, very simple. Just university, we cannot industrialize. We cannot work with many customers, because we should concentrate on the research. So in a sense, as a Biofoundry, service, we set up this kind of companies, and we are closely working together, university and this kind of the company working very closely, and depending on the target, or depending on the project, university work or this kind of company work. So in a sense, this is a very, small, flexible companies. So actually, company looks like this. We have a very basic fundamental, equipment, and this year we are putting a lot of money to the to set up automated systems and so on. So like this is a company, and actually this is a slide, and the universities here. We had a GBA meeting a couple of years ago. Here, just beside the university, we have this startup company. So in a sense, early stage research in the Biofoundry work, university work with the company, and then if the project become more industrial stage, so then the company shifted toward this startup company or something like that. So like this, we try to set up this kind of framework where universities and the startup company working closely to help the industry. Thank you very much for the kind attention.
Thank you so much, Akihiko. That's fantastic news. Really exciting. Really interesting. I do remember that meeting so well. So,
thank you very much. Thank you very much.
Very, very wonderfully generous host.
Yeah, thank you very much.
Any questions for Akihiko? This is an unusual model because this is a very sort of large-scaled kind of a service business, if you like, but working closely with the university. So it's kind of it's a very interesting development, I think.
That is our trial, because universities should focus more on the research, to write a paper. But the industry claimed us that you are just interested in writing a paper. Of course, we are interested in just writing a paper, but they also need the service, Biofoundry service. So in a sense, I set up the company and to collaborate very closely. So this is kind of our challenge to the set up this kind of framework.
Yeah. Fantastic. Well, look, that's wonderful. We've got in the schedule a small break, comfort break. If everyone wants to take 5 or 10 minutes or whatever feels right, that's great. And then maybe we can come back and have Professor Sang-Yup Lee give us the introduction on the KAIST Biofoundry, maybe in shall we say 10 minutes? Okay, 10-minute break. Okay, 10 minutes, then we'll be back, and Professor Lee will tell us. Oh, Tom, hand up. Sorry, Tom.
Sorry, no, nothing to add. All good.
Okay, if everyone's back, hopefully, maybe we can move forward, please, with Professor Lee, who's going to tell us a little bit about the Biofoundry and KAIST, hopefully.
Okay, good morning, good afternoon, good evening, everyone. Great to see all the new friends. Let me briefly update you on K-Biofoundry. So the vision, even though the vision's pretty much same with everyone, but K-Biofoundry is established to help companies and organizations in their activities of biomanufacturing for the a lot of different chemicals, fuels, materials, drugs, and whatever products they want to. And obviously, the mission will be integrating advanced tools of biological engineering and data science and scale-up technologies for the biomanufacturing of the future. So there are 4 main clusters on synthetic biology in Korea. Korea is not a big country, but still having about 57 million people. And as you can see, this is the capital of South Korea, the Seoul. And there are a collection of number of universities and institutes. And they work on all the common themes of synthetic biology. And this in the middle is Daejeon, where I am, where KAIST is located. And we also work on lots of different technologies and also in particularly applied aspects of synthetic biology for the production of chemicals, materials, fuels, etc. Also down here towards the west, south, is Gwangju, major city, and also the other side, that's the east part, southeast part of Korea, where Busan is located. So as you can see from this map, there are mainly 4 clusters working on synthetic biology. Biofoundry is established within KAIST and KRIP consortium, which is in the middle, but we have a collaboration with many universities and institutes within Korea and also abroad. One characteristic so far was that we did not have nationwide big project on biofoundries. So basically all these 20-some organizations and the PIs within secured their own funding, research funding, to perform related research alone or together. And I would say that adds up to about $50 million per year worth of project on synthetic biology. What we have been working on, probably same as you guys. So basically the problems, 3 major problems to address climate crisis and achieving net zero goal announced by many countries. So we're working on developing high performance microbial cell factories for the sustainable production of chemicals, fuels, and materials. And on the problem of healthcare, especially in the aging society, we try to develop new medicine, microbiome-based therapeutics, and also producing health-promoting natural products, which we are mainly focusing on because, I mean, treating patients is not a good way to sustain the aging society, but rather making them live healthy over the aging period will be more important. So what you eat is very important, and we're trying to design the best nutrition for for people in a personalized manner using a synthetic biology platform. And also food and nutrition part, obviously we are talking about soil microbiome here, and also plant and the soil interaction part, and alternative food, not only the alternative meat, but also other sources for the future of food. And also crop engineering is important to make the lot of these grains available when the climate change is really hitting us. And of course, many other topics are under study.
Great update, and wonderful to hear the amazing vision you have for a Korean biofoundry. We're moving now to Lara. To Yue is on. Are you around? Okay, great.
Yes, so I'm sharing. Can you hear me?
Perfect. Oh, Mark, yeah, of course. Yeah.
Yes. Hi, Paul. Just a second. So do you see my screen?
Yes.
Yeah. Okay, so I'll try to make full screens. It's very lucky, my internet. So hi together. Very short introduction to Lara system. I'm from, my name is Mark Dörr from the University of Greifswald. It's located in the very northeast of Germany. So here at close to the border of Poland. And we have a robotic screening platform. It's not in this narrow definition of a biofoundry. It's more like a broad applicable system for many kinds of applications. It's within the institute located at the Institute of Biochemistry at the University of Greifswald, Uwe Bornscheuer's group. And so from the top, it looks like this. So we have incubators and a robotic arm and a centrifuge and microtiter plate readers, 2 of them. So, and then liquid handling station and plate hotels, and all of them are operated by an arm, central arm. Basically, the workflow of this system goes like this. So if you want to have more details on the workflow, then please visit our webpage, lara.unigreifswald.de. And here you see that we pick our colonies. We have colony pickers, so we can grow, induce cells, centrifuge, wash cells, lyser, do assays, data analysis with R, and especially Python right now. And then we feedback. So we do, of course, machine learning. We have collaborations with the Technical University of Berlin and others to establish a closed loop design to make this like a very versatile platform for many, many kinds of research. These are just a few impressions. The full film can be seen in the on YouTube on the link below. But I think I should spend a little bit more time on what we're doing and what we have in mind. So we have this protein screening platform for finding new substrate scopes, altered substrate scopes of enzymes. We enhance enzyme stability, do mainly directed evolution approaches, and screen in the range of 10,000s of variants. With a pre-screening of microfluidics, we can go up to several millions. But right now, mainly we do microtiter-based screening with the platform. And the interesting thing maybe for you is that we go totally open source. So that means that all our devices will be controlled by SILA. So SILA stands for Standardization and Lab Automation, and is an open source and freely available communication standard for exchanging and controlling especially lab devices and exchanging data between them. So what we're trying to do and trying to achieve is building our own scheduler and communicating all the devices with a central scheduler and then a database that stores all the data, all done with open source tools. So, and right now in this week, in the next week, we run our first process on that, and we're very, very excited about it because the scheduler seems to work, and we have most of the devices already integrated with SILA, so that's also very cool. And if you are interested in collaborating with us and seeing the source code, everything is on GitLab. So you will find the links on our webpage, or just write me in a short notion. The idea is that we can achieve this very high enriched data, metadata, by having like an automated process that also adds all the semantic annotation of the data. And that's a very key point to what we're doing to the data, so that the data itself becomes semantically annotated with ontologies. So, and for that, We are partners in the NFDI for CUT, so that's a national data research data infrastructure initiative from the Deutsche Forschungsgemeinschaft. It's a huge community, and within this consortium we are specifying ontologies. So that means if you want terminology, standardized terminology for data, so that machines can understand data much better than they can do before. I think with that, I should stop and maybe answer questions. I think it's more important that you can ask questions. So we have a lot of partners from different universities, and we are funded by the Deutsche Forschungsgemeinschaft mainly. So by that, thanks a lot.
Thank you very much, Mark. It's really interesting, and I love the open source component of your facility and your ethos. Yeah. It's just wonderful.
Yeah. I hope that I can, in a few months, present you, maybe on YouTube, how everything works. So like a kind of preview video of the software we're developing, that you get a more in-depth impression of the capabilities of this software.
That would be great.
Yeah.
Any quick question for Lara or Mark? About Lara, sorry, or Mark. Super. Okay. Thank you so much, Mark. That was really interesting. I think I'm probably next. So just very quickly, I won't take up too much time. Hopefully that's everyone can see that. The London Biofoundry, that's the management team there with David Bell-Marcus-Storch and myself and Richard Kidney. We're based in the Innovation and Translation Hub in White City, a part of London, about 3 miles away from our South Kensington campus, if you've been to visit Imperial. We're in the middle of a sort of what we call a White City biocluster, where there's a whole bunch of different companies and organizations that are very interested in biotech, sort of close by to this west of London. So there's a bit of an initiative ongoing. We have the similar stuff that everyone else has, so there's nothing unusual in our Biofoundry. We've tried to make it a slightly more integrated system that we can do everything from DNA to next-gen sequencing. And obviously we have a number of projects on the go, which is very similar to almost everyone else. But we're a small Biofoundry. We have a core team of about 6 or 7 people. But as you'll see, we have a particular way of operating. So we don't have any core funding, all grant-driven and project-driven with external partners, which is very challenging. And I know that myself and Nicola and other UK Biofoundries are in very similar situations. So at the moment, we're trying very hard to lobby, like signing up to have a much bigger investment into the UK Biofoundry capability. We do focus specifically on entrepreneurship. We have a lot of very small micro seed ideas, companies, young students, postdocs, all not just from Imperial, but from Greater London and also from groups from Oxford and Cambridge and other places that are very interested in starting up companies. So we do kind of specialize a little bit in that. We have projects with academia, and obviously we do try to collaborate with instrument providers. I think there's a great opportunity for Biofoundries to work closely with the new instrument providers and maybe showcase their technologies. We do some training, like Nicola mentioned in her Biofoundry. We typically have 2 PhDs and 4 postdocs that are embedded in the Biofoundry team. And we obviously collaborate with the doctoral training centers, and we help running various automation courses in the CDT. We have some interesting collaborations and hackathons with other partners, and we've also done some training courses for small companies who are interested in getting into automation. We have academic partners, obviously our local Centre for Synthetic Biology, and then we have a strong relationship with the UK Dementia Research Institute. And we have, these are some of our current sort of what I would call more industry, slightly bigger industry partners, some small companies that we've been working closely with. We've done, the last 2 years, we pivoted our Biofoundry to do a COVID response projects. And so we have been very distracted by COVID, and I can't wait for COVID to end so we can get back to our normal activities. But we've been we developed a COVID SARS-CoV-2 testing program, which is now pretty much tested everyone in Northwest London pathology, and Northwest London probably, and we've done over 1 million PCR tests to date. And that's taken a lot of time, so it's now implemented in the NHS and in the Imperial College Healthcare Trust. And we've done a lot of other projects, and some are still being published now. We've worked very closely with a lot of COVID projects at Imperial College. This is an example of interesting beta testing of Syntax DNA printer. This is with DNA script, and this has been really helpful. We're now beginning to produce 200 base pair oligos. We're very excited by having this in-house capability, but it obviously leads to issues around biosecurity and sequence issues around that. But it's certainly proved to be incredibly useful for some of the SARS-CoV-2 work that we're doing. And we've had an upgrade as well, which I think the UK Biofoundry has got a more infrastructure investment, which has been helpful, but we really needed funding for our staff. But we've now upgraded our facility, so we've now brought online some strain cell engineering equipment, some pre-scale bioreactors, next-gen sequencing with a PacBio, and some high-throughput microscopy screening for phenotype screening. Some software development. This is our automated basic DNA. We've just about to publish that. We released that on the web. And we've got some lessons that I kind of shared very, very quickly in summary. From academics, generally it's a shared interest in publications of new research. We didn't want to become a kind of a plasmid provider. Timelines, commitment can vary. It's important, I think, for cutting edge development to have strong academic pull projects. So that's really helped us develop some of the analytical technologies we're working on. Also for research grant income is our lifeline. And we can obviously help support those applications. And then they have access to, expensive infrastructure, and they value that enormously. But academic labs have very different approaches to measurement and reproducible science. So there's some very interesting philosophical differences between how academics approach this, which has been interesting. From industry, we've had many collaborations with startups to large pharma companies who wanted to dip a toe and see what happened. It's a huge effort to set up these collaborations. A lot of resources required, CDAs, NDAs, managing IP, legal frameworks for how you collaborate with industry. So I think that's been very challenging for a sort of academic focused Biofoundry, and it's something that I do think people need to really pay attention to if they want to work very closely with industry. Once you've got it sorted, I think it's very, very helpful, but it does take a lot of resource to do that. And that's it. So I'm happy to take any questions, or we can quickly move on. I'm sure people have got similar experiences. Great. I'm going to move us swiftly forward then. I don't know if David Ross is on the call. Anyone from NIST on the call?
I think you're going to have a hard time with any Americans given the time.
Oh, yeah, fair enough. The next one then is Han-Min Wu from the Sky Biofoundry. If Han-Min's on the call. Oh,
yes,
yeah.
Hello everyone. I'm Han-Min from Samgyongam University of South Korea. So this is a really great time to share the resources and Biofoundries and so on. So my biofoundry is called Sky Biofoundry, or in Korean, we say Seonggyun Biofoundry. And our school is located outskirts of Seoul, just 20 kilometers away from at the center of, let me check, center of Seoul. And there are a lot of biotech companies, including CJ Bio or Daesang or LG Chemistry and so on. So there are many companies around Seoul. So we established our Biofoundry 2020, and actually we gathered the fundings from the government 2018 and 2019. And also we show certain demonstrations. Now we are fully functional, and we are operating certain project that funded by private companies for their interests. And also we are kind of in service for in school and also regional. And also our Biofoundry can be expanded. We are expecting second establishment, and as you heard, sign up gonna be raised big funding. So I hope there also are Biofoundry, which is small compared to K Biofoundry, but still we can expand it and so on. Our Biofoundry is more like a project-based Biofoundry, and we completed a certain workflow of gene cloning and pathway constructions, and also genome engineering and evolutionary engineering. I will show you certain examples today. And those workflows can be done in combination of process modules. So what we call the STEP, so PCR and assembly, and cell transformation, and colony selection, and plasmid isolation. Those also can be done using our robots integrated in our liquid handler. They consist of on-deck thermocyclers and heat control shakers, and etc. So you probably know all machines. Recently, we published cloning method, another cloning method, what we called RoboMoClo, which is robotic assisted modular cloning. And this kind of a hybrid model whole clone, and also golden braid. So which is this cloning method has the features of modularity and hierarchical and iterative and cloning so on. So in our RoboMoClo, we utilize automated PCR and DNA purification methods that benefit for us to design different gene orders and also different gene transcriptional directions any way. And then you can have certain scalability doing your golden gate cloning, and also it's possible with red-white colony selections. And also we have less destination vectors, so we have less handling times. And in the publication, we show certain comparison how when we have full combination of gene cloning A or A and B, and you could have less transcription units before your experiment, then you can do your fully automated or semi-automated procedures for your gene clonings. And we applied it for lycopene biosynthetic pathway, and of course full combinations you have 4,000, but we just for a demonstration purpose we just did it DOE as guided pathway constructions. So only we have 12 combinations of our gene cloning, or we say 12 different pathway units. So we consist starting with the transcription unit of 14, and also we need it's not a full combinations, but we just for applications we built sub pathway units, and then we conclude to building pathway units. So we applied our machines, and we followed all automated or certain part with manual, such as transfer from one side to the other side, and rest of them all fully functional. And then we completed all 12 pathway unit assemblies. Second example of what we could show is engineering. I'm not going to go all the details, but we established also the right combination of process modules, and then we have certain tools for CRISPR-Cas system for Corynebacterium glutamicum in these cases. So we had a functional cytidine base editors and also adenosine base editors, so they are fully functional. So we combine all the process modules, and we could build different guide RNA vectors and strains and so forth. It's not an end of story. We are still analyzing and building sort of parts, and then we could publish it and we can share all the information to you. So yeah, moving forward, we are interested in CO2 fixations and biofuel, and also plant or animal-free chemical productions and so on. So today this is the end, and I can have any questions. Thank you.
Very much. Very nice, very nice publication on your automatic cloning. Any questions?
Min, what do you mean by plant and animal-free chemical production?
For example, if you want to have produce collagen or any protein or the chemicals that can usually extract it from the sources, using the genetics, so engineering, so we could do chemical-free productions.
Okay, got it. So, but you don't mean like not using glucose from plants. You just mean not actually extracting the natural product from the plant itself.
Yes.
Okay, great. Thanks.
Okay. Thanks, Han-Min. That was very nice. The next one is Symbiochem from Rosalind Lefebvre. Symbiochem is up at the University of Manchester in the UK. So I think Ros, I saw her earlier.
Can you see
great.
Screen?
Sorry.
Yeah, is the screen on?
Yeah,
Yes.
that looks good.
Yeah, so Symbiochem is the Manchester Synthetic Biology Research Centre that was formed in 2014 through a very large government grant, and is set up in the Manchester Institute of Biotechnology up in Manchester. So we set up with automation platforms. Okay, slide to move. So that we set up to as a synthetic biology research centre to access chemical diversity based on fine specialty chemicals production. We've created this design, build, test, learn pipeline, which was designed to be agnostic to chemical compounds. And so we've tested this now with a variety of sort of natural products productions, alkaloids, flavonoids, but also going into sort of materials, sort of monomers for materials as well, through collaborations with our various partners. And so we've set up automation, combinatorial assembly, rapid prototyping, chassis engineering, and this is together with responsible research and innovation, so looking at some of the societal sides of chemicals production. More recently, we've managed to upgrade some of our equipment through collaborations with the Royce Institute, which is a materials focused institute. And so we've now got this fully automated production where it combines acoustic liquid handling with PET-based handling. and associated peripheral instruments. As quite a few biofoundries, although we had a very substantial funding at the beginning to set up this pipeline and really sort of set up this biofoundry for chemicals production, our core funding has come to an end, and we now are looking at different models for taking the capabilities forward, keeping core capabilities and the team together. And so what we've done is we've sort of partnered with a future biomanufacturing research hub, which is another large government-funded capability. And we're really trying to integrate the strain engineering capabilities of SynBiochem with the broader picture of how do we accelerate the development and integration of sort of biocatalysts, enzymes, and realize the value of industrial biotechnology at scale. And this hub has partnered with UCL and Imperial, Nottingham, and some scale-up facilities to really try and generate the next generation of sustainable biomanufacturing processes and deliver the capability at scale. So we're trying to bring the focused biofoundry engineering through to that bigger industrial pool. And with quite a lot of people trying to get that collaboration with industry has been interesting. The Future Biomanufacturing Research Hub has managed to get industrial partners to integrate and to try and pull through the technologies. But the initial funding with SynBiochem, it was actually quite difficult at that early fundamental stage to bring in the buy-in with the industrial partners.
Thank you, Ros. That was great. Any comments, questions for Ros?
I guess it was really trying to show that you can gain a certain amount of momentum, but trying to keep that keep the funding going is actually quite an interesting problem, particularly as you change from a core research project base through to sort of fee-for-service type model. It's an interesting transition.
Yeah, we need to sort that out, Ros. In the UK,
we do.
It's not compared to some of our colleagues around the world. I'm beginning to realise we're going to, get well left behind. Okay, if there's no comments or questions for Ros, we'll move on to UC, I think, with VTT. I think I saw.
Yes.
Yeah, okay, great. Perfect.
Hi, I hope you see my slides.
Yeah, if you put them on full screen.
Yeah, I tried actually.
Oh, don't worry, that's fine.
Now. Oops.
We just see a black. Oh, there we go.
Yeah, right. So just let me up. Sorry about this. What do you see now? Because now I see
the title's slide.
The whole slide.
Yes.
Okay, excellent. So yeah, greetings from Helsinki, Finland, up north. So we have 0.5 meter snow here and sun is shining currently. So it's actually a nice winter day. So I will briefly tell you about the VTT Industrial Biotechnology Biofoundry. And so just let's not spend much time to this, but probably VTT is not, you're not so familiar with VTT. So VTT, VTT is the acronym in Finnish for Technical Research Center of Finland. And we are a multi-technology research center, roughly 2.2 thousand people. And we actually, although it's the Finnish research center, so we have a lot of activities with foreign sort of partners and companies. And we are fully state-owned, the Ministry of Economic Affairs and Employment, and we are not for profit. And this sort of shows the strategic choices that VTT has made. So for the focus areas, so work on technological solutions for carbon neutrality, digital solutions, and then sustainable products and materials. And that's where I work in. The here, our research area is roughly 180 people, and it's the VTT Industrial Biotechnology research area. And our goal is to develop efficient cell factories and bio-based production concepts. And really, our focus has been already actually tens of years on yeast and filamentous fungi. We do work also with bacteria, but it's really the main body of our work is on fungal species. And we have, what we try to do is to develop tools that would then be very useful for these organisms. So obviously we are developing bioinformatics modeling, then high throughput molecular biology tools to be able to construct then strains that would efficiently secrete proteins, for example, or then produce through metabolic pathways our target products. And just as an example, so for the tool development, so we have also, we recently developed a method to be able to express genes in a very broad range of fungal species. And this works also in basidiomycetes. So it's because one of the bottlenecks we still have, although we have CRISPR, it's still difficult to start to work with a completely new host organism, because you still need to express the genes if you want to sort of somehow engineer it. And this CESS system has proved very useful here. But then we have this, I mean, many of you have shown similar kind of things that, I mean, there are these pipelines to then robotic things. I have next slide, I think, where there are some details. But then one of the main sort of topics that VTT has worked on also for tens of years is really this upscaling, that we could really make an impact with the bio-engineered microbes that we develop, and so that the bio processes would really produce something useful. So we have quite a lot of sort of upscaling capabilities here at VTT. This is just a listing of sort of instrumentation that in a very short form. So we have a screening robot, it's from Beckman Coulter, and we are actually replacing it with a new one from the same provider this spring, so it should arrive, say, May, June. Then we have this Fluent, which we use mainly for molecular biology related work. Then we have these biolectors, 2 biolectors for sort of testing of small-scale cultivations, and then we have this Amber 250 for these 24 parallel bioreactor cultivations. But then we have over 60 largest bioreactors, all the way up to 1,200 liters. And so this is, as I said earlier, so this has really been one of the main sort of investments we put on, that has been on our agenda. And there's a lot of company interest to sort of also as a service to use this facility. Then I just wanted to highlight, I mean, Nathan mentioned this already early on. So, I mean, one of the things, of course, ways to get these kind of networks to work is to actually do something together. I mean, otherwise, I mean, I feel this GBA is a really great sort of thing to have, and I hope we will sort of come up with very nice ways to collaborate and share information. But then really doing something together is sort of it's rewarding and it's exciting, and then it's a really efficient way then also to transfer knowledge. And so kindly, Nathan, with colleagues, agreed that my postdoc, Matti, is visiting now for 2 months his lab at Agile Biofoundry, and sort of the focus of this meeting, of this visit, is to sort of test how same protocols work on different machines, and then also how we achieve to do the sort of in a harmonized way the upscaling and getting it sort of reproducible. So this is briefly the research plan, and I'm really excited about it. So I think, yeah, so that was
thank you
what I had now. So thank you very much.
Thank you, Jussi. That was great. And I do really appreciate the comment you made at the end there about working together and trying to find ways to do joint, joint activity. I think is a really well made comment. Any comments for Jussi on BTT? Sorry, just a very quick comment, and it relates to our own pilot facilities. I mean, is it expensive to access? If I'm a small company and I want to do some upscaling at BTT, is the business model such that you will make it affordable for small companies to be able to test things, or how does that work?
No, unfortunately, we don't have a reduced price for small companies. So that is some small companies experience as expensive. That is one of the, it's a valid point you're raising there, and it's especially in Finland, where we don't unfortunately yet have a really big sort of biotech industry. So there are these small startups, but then their pockets are not very deep, and then getting this kind of proof of concept things done in bioreactors. So that is one issue.
Yeah. Thank you. That's really helpful.
That's a good point.
Okay. I can't see any hands or anyone. Thank you, Jussi. That was really interesting. I'm going to move on. If you stop sharing, then Human Zhao, who I did see, and I would like to say I really appreciate our American colleagues for getting up in the middle of the night to attend this meeting. It is beyond the call of duty, and so thank you very much. And I'm sorry the time isn't working out this time. Thank you, Human.
So it's fine with me. It's 5 o'clock, and not like Nathan, it's 3 o'clock. Okay. So anyway, I'll give a brief overview of the Biofoundry we established at University of Illinois. We call it the Illinois Biological Foundry for Advanced Biomanufacturing, or IBioFab in short. So we established this Biofoundry in March 2014. So it's almost like 80 years ago. So at that time, it's really just a prototype. We tried to integrate as many instruments together to automate the workflows for synthetic biology applications. So it was a very bold idea at that time, but now, of course, more and more people build a similar kind of Biofoundries. So in the past 2 years, we actually kind of upgraded our system. We added more instrumentations because we realized the limitations with our first version. Many actually is the liquid handling system, right? So we added the two, liquid handlers. One is the Fluent, the other one is the Echo, but also add other capabilities, mainly for eukaryotic cells, particularly like plant cells. We're now doing some engineering with plant cells as well. So we also have the enclosure so that you can work with the BSL-2 kind of organisms. So and of course, I don't think I need to mention all those equipment because you guys all have that as well. But I think what distinguishes us from other places is that we really try to have a fully integrated robotic system, right? Try to achieve the fully closed design, build, test, and loop. And so in the past 80 years, we really try to demonstrate the value of the Biofoundry, right? By performing some proof of concept studies. So the first proof of concept study we did is to synthesize talents, because at that time talents were used as a genome editing tool, right? And nowadays, of course, most people use CRISPR, but talent still has some advantages for certain applications. And at that time, we developed a high throughput workflow for synthesis of talents at a low cost. And then we also tried to use the Biofoundry for pathway engineering. At that time, we collaborated with the ADM and tried to optimize a pathway involved in amino acid biosynthesis. The pathway is relatively simple. It's only 3 genes, and we use the promoters with different strengths and also enzymes with different activities, try to create a combinatorial library and then a screened library to identify the pathways with the optimized flux. And then also we try to use the Biofoundry for metabolic engineering, and this work was done by Tony. I saw he's also on the call, and basically try to automate the workflow for the RNA interference assisted genome engineering effort in Saccharomyces cerevisiae. I mean, all of these are kind of early examples, and then more recently, because we have the fully integrated system, so we try to actually achieve a closed loop for biosystem design, and by incorporating machine learning, because otherwise it's harder to achieve full automation. But what we did is that we also used the pathway, is a relatively simple 3-gene pathway involved in lycopene biosynthesis. And then we use the robotic system to create a library and then analyze the results, and then use the machine learning model to predict optimize the combinations of the promoters to achieve a higher lycopene production. And we can actually achieve this fully closed loop, and I think for the first time. And I think that's mainly because we have the integrated robotic system. And we were very fortunate because I think DOE actually is also interested in this direction, and Nathan, of course, is funded mainly by BITO, while us it's mainly funded by the BER program. So there are 2 offices in DOE are interested in this direction. So in the DOE Bioenergy Research Center, we actually have many PIs work together, try to develop new capabilities for Biofoundry, many of those the design, build, test, and learn capabilities, right? The high throughput screening tools and also the design tools. Particularly, we try to also develop new machine learning tools as well. And we use those tools for the engineering of several non-model organisms. And also more recently, actually, I actually obtained a grant from NSF to set up an AI institute that is really focused on development of new machine learning tools for molecular synthesis. So we also leveraged our Biofoundry, right, that actually can generate a lot of the data that could be used for AI model development. So in this case, we have many faculty actually from the CS department who work together to optimize the catalyst or develop new synthesis planning tools. And of course, as many people are also actually thinking, because how can we make the Biofoundry sustainable for the long term, right? So we are very fortunate that we have funding from NSF and DOE, to keep maintain the Biofoundry and develop new capabilities, but still in the long run, right? So how can we really make our Foundry self-sustainable? So one way to do it, of course, to offer services, right? So that's why in the past few years, we also worked very hard to try to make something that can be generally applicable, and then people can use our facility. So one thing that we actually recently accomplished is to develop an automated workflow for plasmid construction, because plasmid, as we all know, are essential, right, for most synthetic biology applications. So we developed a fully integrated robotic fully automated workflow for plasmid design and the construction. So we have the web interface that can enable users to design the plasmid and then also have the backend software to basically take care of all the ordering and also the operation of the robotic system. And we also come up a very innovative method that is very versatile for construction of plasmid from many fragments. And then also we'll do quality control as well. So hopefully we will make this service generally available, and we definitely are happy to share with anyone in the GBA who is interested in this technology, because I think the more people use it, the better. So currently we offer this service to researchers in the CABI, the DOE Bioenergy Center, and we also want to offer to all the other 3 bioenergy research centers in DOE later this year, and hopefully to everyone. So that's all I have.
Thank you, Karen. That's great, and great that there's a call for collaboration and sharing there. That looks a really very cool bit of technology. Right. Any comments or questions for Human on the floor? I'd be interested to follow that service provision, because as you pointed out, it's a key sustainability issue, an argument about how much resource do we put in providing service, how much we do in providing core research. It'll be very interesting to see your experiences as you develop that service, and what business model you adopt. Will be very interesting to know. Okay, but we're going into the last few talks now. We've got the Singapore Biofoundry at NUS. We've got the Edinburgh Genome Foundry, who didn't get onto the original agenda. And also I've had a request from Shenzhen for a quick update on what's going on there. So if Chulu is on the call, or
yeah,
he's doing this, yeah.
Yeah, so maybe I can just give a quick update from the Singapore side.
Thank you.
Yeah, so maybe let me share the screen. Can you guys see my screen? Yeah, first of all, very, very nice to see everybody. Yeah, it has been a long while. Anyway, so let me just give a quick update on what we have been doing at Singapore site. So essentially the foundry at Singapore is the core facility within our Synthetic Biology Research Center. So essentially it was built to support a number of various research focus that we have in the early days when we first started, looking at biomanufacturing, environmental work, and synthetic genomics, right? Just to name a few. So when we started off, so really is to sort of support projects that has grants that can kind of support the operation of the Biofoundry. But just to mention, more recently we have been focusing more on the biomanufacturing side, really because of the engagement we get through the industry. So later on, I'll say a little bit more. And so like all of you guys, so we have been building software tools, computational tools, and doing automation. But I thought maybe today I could just give you a quick update on the new capability that we have been building. Right. So essentially we now have the EFI, that is being hosted in UIUC. So we are now currently co-hosting it in Singapore as well. So essentially that is to allow us to do more rapid discovery of novel enzymes, through the sort of algorithm like the sequence similarity networks. So I also want to mention about some work that we have done to within GBA software working group. So this is a contribution from a different Biofoundry actually in the early days of the GBA, when we are trying to come together to build some software that different foundry will find it useful. So together we actually built this Python software. We name it as SynBio Python. In fact, if you look at it, it's contributed by different foundry from Manchester, Liverpool, Edinburgh, and then Australia as well, VTT too, right? So that's one example of collaboration where we build software tools together out of GBA. And then at the same time, so recently we start sharing more stuff from the Singapore side. So we have been building biomodeling tools to help people to build models, to study their systems. At the same time, we are building up a collection of strains that we found in Singapore. We are hoping that we can make this available to everybody as well. So it's essentially driven by Ling Hua at Sin City. And the last thing to mention on the capability front, we are starting to focus more on microfluidics, right? Because on the from our needs to really try to increase the throughput, and at the same time to miniaturize the sort of the reagent and the sort of the tools that we need, right? So we are getting more and more interested in building up capability on the microfluidics front. So at the same time, we are also pushing a lot on our public-private partnership. Essentially, this is one of our core activity to bring in funds into the Biofoundry, right? So I believe you guys heard about the Wilma at NUS. That was one of our early establishment of this big private-public partnership. So more recently driven by Wunshan, now we have a Cropper Lab together with Hua Hao. So in short, we name it as HH. So essentially, Hua Hao is a Chinese-based company focusing on bioproduction, right? So I thought it's worth mentioning that in Singapore we have this synthetic biology consortium, which is is funded by NRF to really promote academics and industry to come together. And in fact, through them, we managed to establish quite a few such partnership. And to wrap up, I thought it's worth mentioning this as well. This is a recent collaboration between Wunshan and Paul. Paul, I think you might be able to say more than me. But I think this is a very good collaboration or example of collaboration between Singapore and London Foundry on trying to automate the RNA extraction, in a kit-free manner. So this came up from a need when we start to see all this sort of a shortfall on commercial RNA extraction kit. And then the idea came about, why don't we try to automate using the facility that we have in the foundry to do a kit-free RNA extraction. And I think this is a very good example of collaboration as well between the foundry. And yeah, with this, I will end our updates from Singapore.
Thank you so much, Julie.
Thanks, everyone.
Thank you very much for raising that collaboration, which I should have done. That yeah, that was one of our COVID activities. Thank you.
Yeah, that's right.
Any comments?
Yeah, any comments, questions?
I'm sensing everyone's getting a little tired and we want to get to the end and then we just have a quick wrap up. I'm sensing that because of our different, but very nice progress. And I can't wait to come visit you. Okay, let's move on then to the last two quick talks, which are going to be the Edinburgh Genome Foundry next. This is off the program, unfortunately, but that's fine. So Giovanni, if you want to put your...
Yeah. Let's see my slides.
That's great.
Yeah. So thanks for having us today. It's good to catch up with all of you. So I'm Johnny Strakwadani, I'm co-director with Susan Rosser at the EGF. So for the sake of today, I'm just going to give a brief overview of what we achieved in the last couple of years, I would say, in our foundry, which is really designed to enable scientific community and accelerate industrial biotechnology and synthetic biology in general. And so we have been doing this by effectively implementing high throughput DBTL cycle. We now fairly cover all the 4 steps of the cycle. Recently, we have added through some of the work in my lab, the learning aspect, which I'm not going to cover today, but where we really made some huge progresses in the build and the testing step of the process that really allow us to take on new projects that are more and more challenging. And you probably know what is our project's life cycle. So we welcome customers in our foundry. We do an onboarding step where we go through a number of different things, biosafety screening, as most of you feasibility evaluation. And actually one of the things where we spend a lot of the time, and is much appreciated by our customers, is custom design and preparing their constructs. This is mostly because we are able now to maximize their success in the lab. And we are recently developed a platform to handle all this information that we are delivering in the next couple of months or so. The assembly, planning, and let's say design has been completely automated. We have lots of tools that allow us to check in silico everything is working as expected. And of course, we have very strong manufacturing capability, mostly based by robots, as you know. And we also have now a very efficient QCing step that allow us to deliver error-free DNA. So how we achieve that? Pretty much the design part is demanded and delegated to the software branch of the Foundry, where we have developed a suite of almost 40 tools that allow us to go through the whole workflow and provide the information to the designers and the people in the lab on how to troubleshoot, design, and evaluate the work we do. It's all open source. You can, and you probably have already used some of our softwares, but this suite is really expanding as we speak. And you probably have seen our Foundry is one of the most automated out there. Is currently able to do 6,000 constructs per year. This is always a number that we strive to move up to some extent. It was funded by an initial commitment from the government of roughly 5.4 million, and now is really paying off the dividends with the stuff we are doing. So one of the things we introduced very recently is a new pipeline that allow us to deliver error-free, let's say, DNA to our customers, and we do so by validating all our construct now using nanopore sequencing. And we come up with a protocol that actually allow us to basically give whole plasmid results for roughly 8 pounds per construct, which we tested somehow the internal market, I would say, and it's quite well received because it will allow us to, and the customer, to save a lot of time when it comes to troubleshooting. And we have a very nice protocol in place and analysis pipeline that gives us like 100% sensitivity and 98% roughly precision. So we are very excited about that. And 2 of the software that we are have developed in the Foundry, thanks to Peter Weg, is going to be released very soon, and we should have a paper shortly. So but what I want to really highlight now is that we are really stepped up in the testing part, and we are one of the few in the world to have at this point in time a Berkeley Lights beacon platform. It's a really revolutionary machine, 2.1 million investment from BBSRC to really scale up testing in mammalian cells. And when I'm really talking about scale up, it means moving from months into hours and days. So really dramatic changes on what we can do in our Foundry. And this goes by different type of applications, ranging from antibody discovery, cell line development, gene editing, and obviously cell therapy development. The nice thing is that being a microfluidic device, so we can measure a single cell level, and in real time, we can do cell culture in a closed and sterile environment in a very massively parallel way. And we can actually select the single cells for doing off the machine experiments, like, for example, single cell RNA-seq or whatever other type of phenotyping you want. We currently have different workflows for cell line development, antibody discovery, and single cell RNA-seq, but we are expanding this in strong collaboration with Berkeley Lights. And we are very excited by the type of application we can enable with this piece of equipment. So in terms of portfolio, very recently, we really scaled up the complexity of what we can do in the lab. We started, let's say, since 2019 with a COVID project to design a new next generation ELISA testing system. And we're now moving into more standard assembly approaches. One of the projects, Cyanosource, is aiming at building a library of mutants, modular mutants for cyanobacteria. There's been quite an effort to make it to work together with the Erlang Foundry, which is another good example of cooperations between our facilities. And then I'm glad to see so many other foundries moving into the enzyme field, and we are really interested in therapeutic enzymes. This is part of the work we share with my lab. And we are trying to build large libraries for treating metabolic disorders in the order of 1,000 enzymes that are fully designed and characterized. And recently we are scaling up for cell engineering. We are gonna work on a different type of aspects, ranging from synthetic lethal screening in high throughput fashions to engineering cell lines for bioproductions and biologic productions. And this has enabled, I think that's a key for all the biofoundries, is that having these capabilities and the abilities to do this type of project has enabled us and our customers to attract a substantial amount of funding from different agencies in the UK, and also has attracted a lot of industrial partners that we are working with, and we are in very deep conversation with Fujifilm, who is already working with the University of Edinburgh, Sartorius, AskBio, and others, just to name a few. So the problem, I think, is that sometimes it's very difficult to enable this type of project at a scale that is sustainable for PIs in an academic environment, and is also challenging to meet all these demands and be competitive for an industrial application. So I think there's also something to discuss in this regard. Last thing I want to say is that all this has been possible thanks to our amazing team. I want to stress a lot that this is an interdisciplinary team of scientists. It's not just technicians that do the work, but they actually do active science, and we help the customers delivering their own project. Renos Pragut is also here, but also Peter Beck, our computational biologist, and our research technologist, Sophie and Gaynor. We just appointed a new technician, and we have an opening soon, I think, for a computational biologist and another research technologist. So with this, I'll close. Please do get in touch if you're interested in working with us. Renos' email is here. Mine was on the first slide. If you want to talk about software, please do get in touch. All our software, or most of it, is already online, open source on GitHub and our website. And we also have a very active Twitter account these days. So please do get in touch. Thank you.
Thanks, Giovanni. Very nice talk. Yeah, some chat. Nice to hear about interfoundry work. I think that's a really got some examples of real work ongoing, which is great, and we really need to expand that. Okay, I think we'll, just for the sake of time, to give us some time to chat at the end, I'll probably just move on to the last talk. I was sort of last minute request from Shenzhen. I'm not sure who's delivering it. Whoever it is, please declare.
Hi, so Professor Freeman, this is
Yes.
Tom. Hi.
Okay,
so,
yeah, great.
Well, let me share my screen. Okay. So that looks good?
Yes.
Okay, so thanks everyone and Professor Freeman for accommodating our last minute request to provide a quick update on the Shenzhen Biofoundry. So I'm Tom, and I give this talk on behalf of Chen Li. So just a quick reminder, so this foundry will be built in the southern China. So it's Shenzhen is a city like close to Hong Kong. And this is a rendering picture of the foundry, which will be hosting a new building. And I want to point out that our institute, the Shenzhen Institute of Synthetic Biology, which is part of the Chinese Academy of Sciences, will be located very near. So it's going to be in the same cluster, and we will have basically very close access to the foundry. And some basic fact about the project, it started in 2017, and right now it's 2012. So we are in the process of the installation and commissioning. So it's going to be finished in the next year. And it will be hosting the building, which is 14 floors. And we have 4 highly automated laboratories. And the building is still under construction, but you can see it's pretty much finished. So the completion expected in July this year. And currently we are constantly recruiting, and the target will be 80 engineers, biologists, and admin. So for these 14 floors, we're gonna hosting 27 robotic work cells divided in 4 platforms targeting DNA synthesis and analysis, model microbial chassis, including E. coli and yeast, for both DNA cloning as well as the like cell factory engineer as well, and as well as the non-model chassis, which is more versatile, like the anaerobic, gut microbiota, some cell-free system, mammalian cell, plants, etc. And as well as a synthetic phage. We basically like to both engineer those for targeting antibiotics resistant strains, as well as creating synthetic phages as well. So those red ones, I'll provide a little bit more details on the progress. And also because the new building hasn't been finished yet. So temporarily, we are prototyping those lines in the Industrial Innovation Center for Engineering Biology, which I think is about several miles from the new building. We are still under construction, but this innovation center is also newly, it's a existing building, but recently renovated. So basically hosting those startup companies or spin-offs of these institutes, trying to integrate the basic research and commercialization in a single space. So because we are prototyping the automation work cells in this building, so we think we are also experimenting with the new models for serving the industry, also as a few speakers mentioned, for the sustainability after the government investment, of course. So we can see there are these new spaces for startup companies and the prototype lines we are putting into it. So these prototype lines pretty much targeting for DNA assembly at a scale of 3 to 10 kb, which we think basically, as also Professor Zhao mentioned before, this is probably one of the most popular requests from both the academia and the industry. So these are the prototype lines, and here are the workflows and associated the instruments. So this is actually a picture from a recent issue where Professor Huiming Zhao lead. So basically, we are describing the this prototype line we are creating. So once the full line is built, we are targeting about 7,000 contracts per week. But currently, with this prototype line, we can making around 1,000 per week. So another thing I want to mention is like in this building, we are also trying to prototype a fully automated anaerobic chambers hosting all the robotics. I know Lonza is also creating one, but that one is more engineering in Australia. This one is more like isolating, trying to do the so-called ultraomics, trying to isolate as many as possible the human gut microbiota. This is because we have microbiome center as well as this request from both startup companies, Bio, as well as big companies, Cofco. So they are, because we are replacing the antibiotics with those more like ecology engineering stuff. So we want this as well. I do really want to point out, like when we build this fully automated anaerobic work cells, there are some engineering challenges. For example, the QPix, which is a commonly used colony picker, is too big to fit. So we have to, so basically our engineer team doing this customized like plater, which we are creating the consumables as well as just mount them on these robotic arms and the pipetting and for this plating, and as well as the microfluidic system, so where we can get single colonies based on this Paulson or limiting dilution principles. So that's some engineering capabilities we have, especially when we want to creating this new kind of automated work cells. So I think that's all I have. And thanks a lot for your time and attention.
Thanks, Hong-Si. That's looking amazing. The building and everything looks fantastic. Do you know, can you just say what scale of investment has gone into the Biofoundry in Shenzhen?
I think 700 million Chinese yuan. So I think that's about $100 million.
Fantastic, amazing. Is there any strategic focuses for the Biofoundry is it all on biomanufacturing, or is it, I mean, you showed you've got many different areas that you're going to explore. Is there anything that's higher agenda?
Right. So I think there are a few projects. The first thing for scientific, that's more like the synthetic genome, because Professor Junbiao Dai is in here. So before they have synthetic yeast, right now they are doing the synthetic plant genome. So that's where we also have a work cell going to be designed for the moles, I guess, the moses. So they are doing for that. And as well, we have, I think Jay Kisling has a center here focusing on synthetic biochemistry. So there are a few plants and products we would like to put into the microbial chassis for production. And there you have the enzyme pathway stuff. And another thing is the microbiome. The microbiome we are trying to basically automate. So yeah.
Okay, thank you so much. That was great. I realized we've run massively over and we only have literally 10 minutes left, but I would like to use that 10 minutes just to open the floor. I did put down a few questions. We won't get through all these questions, but maybe, I guess the most important thing is how do we move forward, and what areas could we work on, or whatever anyone wants to comment or say. But it's been really fantastic to see so much progress, and gosh, we have been missing these meetings. You know, it's wonderful. And next time, I hope we can all really get together and really, you know, really work closely together. But so I open the floor for anyone who wants to comment or kick off. Lars.
I guess just one comment, given short time, etc. Let's get together. I think that's, I mean, we really need a few days to talk together to enable this. I mean, each of these points are many hours of discussion. So I think that would be the first part of the agenda. So
I completely agree, and I think we take this offline and we work hard to identify hopefully a place where we can all come together, and someone willing to host that. But I 100%. So thank you for saying that. That's I agree with you.
I'm happy to go to the Novo Nordisk Foundation and ask them to, I mean, they have grants, but maybe taking an offline of just, I mean, it might be 150,000, but at least allow us to fly in a number of people just to do it quickly. I can do that as soon as
I agree. And also Nathan's put on a chat, Vince Martin, obviously we got very far with that meeting and almost happened, and then, you know, COVID happened. So maybe we can go back and revisit that. But my feeling is we need to meet somewhere, anywhere, that everyone can meet, you know, that everyone can travel easily, you know, with visas and whatever. But I hope so. Thank you for raising that. I mean, yeah, we're very short on time. Has anyone else got any other comments and things that want to, bring to the table?
I mean, I guess the only thing that I would say is don't wait for the next meeting like this to ask your questions. Feel free to reach out and ask for support or offer your assistance to others.
Great point. We don't need to meet form. I mean, we're all there on the web. Everyone knows pretty much everyone. And if there are anything that anyone wants to contact, I really support that. I mean, the more we can work together, that's the whole point, really. And support each other, definitely. And there's so much great stuff going on. I mean, this meeting's been really terrific, actually. I think we need a meeting. I think that's probably the final conclusion of this whole. So rather than prolong...
Paul and Karen, maybe just one procedural question. Are we going to make the recording of this available, or where is this going to go?
Wherever you feel. Oh, there we are. Wherever you feel appropriate. We were going to make it available to the wider colleagues who couldn't come today. You know.
Yeah, I think it would be very helpful for them to see, because there's so many good presentations and good updates.
Shall we just circulate it within the GBA wider community? Yeah. Okay. Great.
Yeah. That'd be great.
Fantastic. We will do. Yeah. I mean, I'm just so excited. Oh, sorry. We've got a hand from Mark. Is it Mark? No.
Yeah. Hi.
Oh, yeah. Mark, yeah.
Can you hear me? Is it working? Yeah. I'm just wondering. Speaking of infrastructure, do we have a kind of repository or something where we can at least have like a short overview of what people are working on, what techniques they offer, that it can quickly pinpoint and select the one collaborator and partner? Is there something available?
No, it's a great idea. Me and Karen can take that offline. I mean, what we'd probably ask is if sort of a little couple of sentences and then a link to Lara or to EGF or wherever, you know, that illustrates those. We can definitely do that on the website to provide a kind of resource, at least a sort of a single place to go to see. We can try and put that together. I mean, it would take everyone to help us do that, but we'd be happy to try and do that. Because we have got control. So that's a great idea. Thank you.
You're welcome.
Anything else anyone wants to say, contribute? So I got the take-home message I've got is we need a physical meeting as soon as possible, and we're going to take that offline and work up that and have a 2-day meeting where everyone can come together and have posters and have a whole proper discussion sessions and everything. Second point was, can we try and have some sort of web-based resource, at least a link into other people, into where people can find information about particular activities within the wider community, the GBA community. So I think that's a great idea, and I'm really happy to take that on. Is there anything else anyone wants to mention or finish? I mean, maybe we just leave it at that. I'm happy to leave it at that, but I realize people have different hours and things. But it's been a really super meeting and amazing. Okay, great. Well, why don't we spend all our efforts in trying to arrange a physical meeting like the wonderful one we had in Kobe, which I still remember with great fondness. Akihito, I know you're still on the call, but the sake tasting was was very memorable, and I think we need more of that. Great. Okay, I'll just wrap it up. Thank you so much to everyone, and I really appreciate all the time zone. And we can. I'm just going to put full efforts now in working with there's a steering group with the GBA, with membership. We can look at that and revise that. Some people have left and some people have joined, and we can take that up and try and organize this physical meeting as soon as possible. Okay.
Thanks again to Paul and Karen for organizing and getting the meeting going.
Yeah. Thanks a lot.
Thank you very much.
Thanks, Paul.
Always good.
Thank you very
Thanks.
much. Yeah, thank you very much. Good.
Thank you very much.
Thank you.
Bye-bye.
Bye-bye.
Bye, everyone.
Thank you very much.
Bye. See you next time.
Yeah.
Bye-bye.